there, and welcome to our Ask Me Anything session on proteomics. I am delighted to have you join us here today, and as the name suggests, this session provides you with the unique opportunity to ask our distinguished guest absolutely anything about his work, background research, science, and innovation in the field. Before we begin, I just want to highlight a few important details. Firstly, this is a live session, and we are thrilled to see so many participants joining us today. Many of you have actually already submitted your questions when you registered, and we really appreciate your support and interest in the session. But don't worry if you haven't submitted your questions yet, because there's still time. You can add your questions to this live session by entering your question into the box on the right-hand side of your screen and clicking send. So if you have any questions for our speaker, please do share them with us. We've got a team in our virtual backstage who will be ensuring the questions are appropriate, so please do keep them relevant. And we'll do our best to address as many questions as possible before the session concludes. Whilst you're listening in, be sure to explore the literature section as well that you'll see on the platform, where you can find some fantastic resources available to download for free. If you do come into any technical issues throughout the webinar, just click the chat icon in the bottom right corner of your screen and our team will be sure to help you out. Once again, welcome everyone to our Ask Me Anything session, and today, without further ado, it is my great pleasure to introduce our expert for the session, Dr. John Yates. John, how are you doing?
I'm fine, thank you.
Perfect. So John, could you give us and our viewers a little bit of an introduction to who you are and what you do?
So my name is, my name is John Yates. I'm at the Scripps Research Institute in the Department of Molecular Medicine. I also have an appointment in the Department of Neuroscience. And I've been working in the area of mass spectrometry-based analyses of proteins since 1980. So for a long time, long before it was protein biochemistry was called proteomics.
Mm-hmm.
And I started my career so I went to grad school at the University of Virginia. I did a postdoc at Caltech in the laboratory of Lee Hood.
Mm-hmm.
And Lee was a pioneer in the development of microchemical instrumentation to sequence and synthesize proteins and then sequence and synthesize DNA.
Mm-hmm.
And his lab was, in the 1980s, was like the hottest lab in the world at the time. Very exciting place to be. Then I started my career at the University of Washington
Mm-hmm.
in a new department called Department of Molecular Biotechnology. Excuse me. And from there, I was there for about 8 years, and then I left and came down to San Diego, where I'm at the Scripps Research Institute in La Jolla.
Fantastic. We recently spoke with Lee, actually, so small world. And could you let us know how you first became interested in studying proteomics?
So the long story is, when I was in college, I intended to go to medical school.
Mm-hmm.
And I took organic chemistry, which is required of all pre-med students, and I absolutely loved organic chemistry. Did really well in it. And when I didn't get into medical school, I thought, well, organic chemistry is an option. So I intended to take another year, study some more chemistry, and then because I was a zoology major
Okay.
in college, so that's animal biology. That's what back then the field but the field of biology was divided into animal biology and plant biology,
Mm-hmm.
and the animal side was called zoology. So I spent another year with an intention to reapply to medical school. But the university had just gotten a new mass spectrometer, and so I was very intrigued by this. And they also had a new faculty member, assistant professor, who'd come out of Klaus Biemann's lab, Robert Anderegg. And
Mm-hmm.
so I joined Rob's group, and he was using Biemann's chemistry to sequence peptides. And that was a very elaborate process of derivatizing the peptides to make them volatile, and you use GCMS GC to separate the peptides, and then they enter into the mass spectrometer, and you can sequence them. And you're generally sequencing peptides that were reasonably small, so 2 to 3 to 4 amino acids in length. It was clear to me there were some changes coming in the field of mass spectrometry. Fast atom bombardment had been developed, where you no longer needed to derivatize peptides, and so changes were coming. So I decided I'd go to grad school, and I wanted to go somewhere with a a bigger program.
Hmm.
And so I went to the University of Virginia in Don Hunt's laboratory and worked on protein sequencing by tandem mass spectrometry.
Nice. Very, very nice. And I think because we have quite a varied audience with us at the moment, it would be really beneficial if you could give us your kind of overarching definition of proteomics.
So proteomics. So back when I was starting, it was protein biochemistry, and the goal was to sequence a single protein and
Mhm.
then study that protein. And one of the things that emerged towards the end of the 80s is that electrospray ionization came along. That allowed us to interface liquid chromatography with mass spectrometry, which had been a long sought goal. And also emerging in the early 1990s, so late 1980s, the early 1990s, and you probably heard this from Lee Hood when you talked to him, was that the Human Genome Project was starting up.
Mhm.
And as a way to get that to get buy-in across the scientific community for the Human Genome Project, the people in charge agreed that they would sequence all these other organisms, like yeast of Saccharomyces cerevisiae and fly and worm and mouse, and they would be model systems by which people would try to understand the functions of the genes that would eventually be discovered in the human genome. I mean, it made a lot perfect sense to do
Mhm.
that. And it was kind of a place where we would work out the technologies to sequence large genomes in preparation to do the human genome, which would be a lot bigger. And so as that emerged, people, my lab in particular, discovered that we could use that resource to interpret tandem mass spectra of peptides. And what that did is it turned a process where you sat down with a calculator and you tried to figure out the amino acid sequence of a peptide from the spectrum to an automated process that was reasonably fast and actually surprisingly accurate.
Mhm.
And so that method grew in power as more and more sequences started to enter into the sequence databases. And so it got to the point where by 1997, the Saccharomyces cerevisiae genome was completed. And so one could do very powerful studies uh, in yeast using mass spectrometry as a way to figure out what proteins you were looking at.
Mhm.
And what so in the initial days of proteomics, um, before it was termed proteomics, people were using gels to separate proteins and cutting bands and then trying to identify those proteins. But it was very clear from um, the 2D gel electrophoresis was very popular, and it was very clear that you could see large numbers of proteins on these 2D gels, and so people assumed it was like the entire proteome that you could see. And as a result of that, the term people started to think about, well, rather than just analyze one protein at a time, we have this capability to use these 2D gels to look at all the proteins, all the changes that are going on as a result of some kind of disease perturbation to the system. And prior to mass spectrometry coming along, the only way you could study those spots on the 2D gel was if you had an antibody that lit up a particular spot, then you knew what that antibody bound to. You could be able to identify that protein. And um, you could run a whole bunch of gels and cut out those spots, the same spot, and then run them on an Edmonds sequencer.
Mhm.
But that was a huge amount of work, and so it could take you up to a year to actually figure out get enough sequence information from that protein uh, to clone the gene. And mass spectrometry came along, and all of a sudden you could just pick spots off these things and identify them very very quickly. One of the things that my lab did is that I didn't have the patience to run 2D gels or even a single dimension gel for that matter. So I thought, let's find a way to get around gels and just analyze protein mixtures directly by mass spectrometry, which was enabled by the process of tandem mass spectrometry
Mhm.
and the database searching software that we had developed. And so now we could identify proteins in a mixture, and they kept getting uh, as methods improved and instruments improved, the complexity of those mixtures got greater and greater and greater over time. And so that really enabled one to think about, which is where the proteomics term came from, is to think about not just looking at single proteins, but looking at systems of proteins or even whole cell lysates and so forth.
Hmm.
And so the story of where the proteomics name came from is actually quite interesting. There's a couple of grad students down in Australia, Mark Wilkins in particular gets credit for this,
Hmm.
decided that protein biochemistry needed a new name that would be fancy like genomics. And Mark was given a talk at the Siena 2D gel electrophoresis meeting around 1994, maybe.
Okay.
And he stood up, gave his talk, and used the word proteomics, and it just caught fire.
People were
Everybody,
interested.
yeah. So that's sort of my definition of proteomics. And it's got different definitions for slightly different definitions for people, but basically it's looking at systems of proteins and trying to understand functions, the physiological roles and functions of the proteins.
Fantastic. No, thank you ever so much. So we have already had quite a few questions come in into the chat box on the right-hand side of your screen. And of course, everyone who's watching along, if you've joined us slightly late, do make sure to pop your questions into the box on the right-hand side of your screen and click send. And we'll be getting through as many as possible in the time we've got available. So a lot of people are having a bit of a chat about the database searching software you developed that you just mentioned in the last question. People are wondering if you could tell us a bit more about that.
So the prior to mass spectrometry coming along, one of the ways that you could search databases, sequence databases, was you could use a short stretch of amino acid sequence. And one of the most famous algorithms for that is called BLAST. But prior to that, there were other algorithms like FASTA and so forth. And so these would you could either do an exact match to a sequence, or you could do a sequence similarity search, and so find similar sequences. And what we did with tandem mass spectrometry data is encoded in a tandem mass spectrum is a sequence. And so if you know how to derive that sequence from the fragmentation patterns that's in the tandem mass spectrum, then you could write an algorithm to create an interface sort of between the sequence in the database and then the sequence that's represented by the fragmentation patterns.
Mm-hmm.
And so my lab was the first to show that you could do that. And there have now been like 35 or 50 different implementations of algorithms to do the same thing. And then people have allowed for sequence variations and unspecified modifications and so on and so forth. But once the proof of concept was done and we understood how to make this work, we could then start adding features like look for modifications. Modifications aren't specified in the sequence databases. So something like phosphorylation on a peptide, you might suspect that it's there, but there is no information, at least at the time, there was no information in the database that this site in a protein sequence was phosphorylated. And so it would not be represented in the database. So we developed ways to go searching in an unbiased fashion and without any prior knowledge where phosphorylation might exist. And this gave rise, the ability to do this gave rise to the field of phosphoproteomics, where people have been generating massive numbers of phosphorylation sites in proteins.
Incredible. I think that's super, super interesting. But actually, so as is the nature of an Ask Me Anything session, we've had several different questions come in. I've picked another one. Someone here is asking you about proteomic diagnostics to better understand and treat things like long COVID-related illnesses. Do you know anything kind of about that kind of area?
Tiny bit. Mostly from my role as an editor, where I see papers come in where people
Mm-hmm.
are looking at blood from patients to try to understand what are the signatures for short-term COVID versus long-term COVID and things like that. Now, people are and it's going to take a while to sort this out. So what are why are people getting long COVID? And I've had a couple of friends that had very bad cases of COVID and had suffered from long
Hmm.
COVID. Took a while to get over it, if they actually did. But proteomics can be used to look for signatures in blood that might be signs of that you have long COVID, and it will take some effort to understand what are the other changes going on. Like people talk about brain fog, and there are
Hmm.
some cardiac issues that have emerged. So studying those particularly in people, are a lot harder because not easy to get a brain biopsy from somebody, so you can go and check on
Ooh.
them. And also not easy to get a heart biopsy too. And so one of the things that we need to do is find appropriate animal models that'll model this disease, and then use that as a way to try to study it and find out what are the molecular changes, and then how we can reverse those molecular changes that are occurring. Hopefully that makes sense.
Absolutely. No, very, very interesting. Thank you very much. Um, a lot of people have been asking for your thoughts on AI and how you see AI influencing the field of proteomics.
AI is already influencing the field of proteomics, and there
Hmm.
have been some very clever uses so far. Um, it hasn't been probably at the same level as what you might think about with ChatGPT
Yeah.
and people writing high school term papers or something. But one of the more interesting things, I think, is that people have been trying for a long time, my lab included, is to try to predict the abundances of fragment ions for a sequence. And there was a group in Germany that used an AI algorithm to come up with a way to predict what those fragmentation patterns. Actually, there were two groups. There was one in Germany and then one in Belgium that did this. And they developed algorithms AI algorithms that would predict and fairly accurately predict the abundances of the fragment ions. So you have to learn how the algorithm has to learn how the amino acid sequence influences where and how often a bond in the peptide will fragment and create more of a fragment ion. And that's been one really nice success of using AI in proteomics. Another one has been predicting HPLC retention times
Yes.
on
Hmm.
using AI. People have been doing this. When I was in grad school, there was a paper that tried to predict retention times. But now we actually have uh, very, very large amounts of data. Back then, they were kind of running peptides and seeing where they came out and so forth, and then trying to infer from there. But now we've got massive data sets in the Pride database over in England that people can use to train algorithms to predict retention time. And that's now gotten to be very accurate and very robust.
Nice, fantastic. Um, a kind of similarly themed, I've seen some questions here come in about data being uploaded to the cloud. I know it's a bit of a big topic. Someone here is asking, is it becoming a bit of a concern for you, or is it the kind of the cost that's a concern? A lot of people asking about patient data in the cloud with proteomics and kind of your thoughts on that.
Yeah, patient data in the cloud is a little bit out of my expertise because there every country has different laws surrounding patient privacy data,
Hmm.
and um, in the U.S. we have these things called the HIPAA laws, and so we have to worry about that. And so this is way above my pay grade.
Hmm.
But I will tell you that one of the hottest resources in science right now is the UK Biobank.
Yes.
And so you have not only do you have massive amounts of clinical data on patients, but you have lots and lots of samples from patients. And so anybody doing proteomics in particular, but also genetics, would like access to that data and those samples to try to understand and study diseases.
Hmm.
The cloud is so at Scripps we've always had so I always say that I have two favorite perks at Scripps. So one
Okay.
is free parking and the other is free computing.
There you go.
So Scripps has always had a large number of people interested in structural biology, so protein structures. And as a result of that, they've always had excellent computing resources. When I first arrived here, they had a Cray supercomputer, for those of you old enough to remember. It was subsequently replaced by Silicon Graphics computing cluster, and now we've got one of those, just one of the standard, probably Dell massive
Huge.
clusters with 6,000 CPUs or something like that.
Hmm.
And Scripps has for a while has been kind of sort of watching what's the cost of having our own versus the cost of using the cloud for doing large scale computing,
Hmm.
and we still haven't shifted. And for me, paying to do things in the cloud versus things that are free, it makes no sense.
No brainer.
So it's hard to beat free for the time being.
Hmm.
And the data storage is cheap, so we buy our own storage and all that kind of things to maintain it here.
No problem.
But it is a at some point it's going to become cost effective to do things more in the cloud.
Yeah.
And there are increasing numbers of computing systems, particularly for proteomics, that are operating in the cloud. You upload your data and you process it up there, and
Hmm.
so it's coming along.
Nice. Lovely stuff. So we've also had quite a few people who are interested in anything you're working on currently. Are you able to tell us anything that you're doing right now?
Sure. So we're interested in what I'm particularly excited about are three things.
Hmm.
So one is single cell proteomics, and that's kind of all the rage in the field as well. The second thing is what I like to call 3D proteomics. So it's looking at the protein structures or conformations using a method called chemical footprinting, but doing so on a proteome-wide scale, and doing so inside cells and inside tissues and so forth. And so the idea is to be able to look at how diseases might be transforming the structures or conformations of proteins, and does that lead us anywhere? We're also trying to apply these same strategies to some diagnostic questions as well. So there are diseases which result from protein misfolding. Alzheimer's disease is, of course, the most famous. So can we use the methods that we've been developing as a way to try to understand how protein misfolding occurs? And ideally, you'd like to be able to pick that up really early in the process. I mean, I was just reading the New York Times just had this big article. So Perdita Barron in the University of Manchester has been studying this woman who can smell Parkinson's.
Yes. Hmm.
And so the really interesting thing about this woman is that she can smell it on patients long before they start showing any symptoms.
Yeah.
And so ideally, you want diagnostics that can do that, that can pick up things, particularly in these protein misfolding diseases like Alzheimer's. By the time you realize somebody has Alzheimer's, it may be too late to treat them.
Hmm.
So you really want to come up with diagnostics that tell you very early when somebody has some disease. And so that's one of the things that we're looking at is whether or not these protein misfolding signatures can be picked up early in the process. And then the third thing is so top-down native mass spectrometry for looking at how intact proteins can be used to study biological functions and diseases and so forth. There's a term of art that we use in the field called proteoforms, and these are you might think that one gene produces one protein, but it's actually one gene produces many proteins, because they're not only can be different sizes because they get trimmed back and forth depending on the type of function they have, but they also may have different modifications on them, which in effect creates a sort of a slightly different protein and can also have slightly different functions.
Hmm.
And so trying to sort that out, so when we have a different proteoform of a protein, what's it doing? Why is it different? And do we get those proteoforms? Do they happen on proteins which are now have aberrant function, that are not doing the right thing?
Hmm.
Uh, and in fact, there's one of the proteins implicated in Alzheimer's is a version of this tau protein, and it has a particular phosphorylation site that's been implicated in the disease, and people use that as a diagnostic marker. So there are many different, because there are many different because tau gets hyperphosphorylated in the disease. There are many different
Hmm.
proteoforms of tau, the tau protein. So we'd like to use mass spectrometry and native and top-down methods to look at that. And that's a field that's been growing too. It's finally got to the point where I feel safe to jump in. It was not an area that I wanted to develop per
Yeah.
se,
Hmm.
because it required very big, very expensive mass spectrometers, and
Yeah.
no longer does. And so
Hmm.
it it's more it's getting more democratized, I would say.
Fantastic. And do you know what? That was a brilliant answer, because we've had so many people asking questions about Alzheimer's. So that actually kind of answered two in one. So we've also had someone here has popped a question into the box on the right hand side of your screen. And she said she's been thinking about proteomics and personalized nutrition, dietary recommendations. Is it possible to link them up?
Well, probably. Um, there have been some efforts among some groups to try to use, in particular, sort of the one of the leaders in this field is this guy at Stanford University, Mike Snyder, and
Mhm.
he's come up with what he calls IPOP, and
Yes.
it's individualized personal omic profiles, and he's using all these different profiling methods, including proteomics, but also metabolomics, to try to monitor people's health.
Yes.
What's been really interesting coming out of Mike's work is that he has a set of people who are wearing monitors, and they're getting blood work done on a weekly or
Hmm.
a daily basis, something like that. And is the sort of things that we think are normal numbers, like say 120/80 for our blood pressure,
Hmm.
may not be normal for everybody. And so that's where people have taken a large number of blood pressure readings, and that was the average. And not everybody's average. And so, and not everybody has the exact same body temperature. Some people might run a little
Hmm.
bit hot, some people might run a little bit cold.
Hmm.
And so these are sort of things that Mike's pulling out of the studies that he's doing, using trying to combine the molecular profiling methods with these type of measurements.
Incredible. We
So
actually also spoke to Mike Snyder recently on an Ask Me Anything session. So his work is fantastic, because he's also at Stanford.
yes, he's at Stanford.
Perfect. Lovely. So just to let everyone know, we're about halfway through this Ask Me Anything session. So just to reiterate, this is obviously a live session. If you do have any further questions, pop them into the chat box on the right hand side of your screen, and we'll be getting through as many as possible in the remaining time that we have available. Someone called George here has asked, what does a typical day look like for you in your role?
Well, I usually get to Starbucks somewhere between 6 a.m. and 6:30,
Not
and I
sponsored.
and then I get into the office. And so I'm on the West Coast. And one of the things I learned when I moved to the West Coast probably 35 years ago when I went to Caltech, is the world, at least the USA, kind of operates on East Coast time.
Hmm.
And so I kind of stayed on East Coast time. So I start very early, uh, get in, I have a bunch of emails to clear, and I'm a journal at an editor. I'm an editor at a journal, and so I may one of the things I may do is I may start my day going through what articles have come in and assigning them to the associate editors. Um, I one of the things I like to do early in the morning is write, and
Hmm.
so I may write because it's still quiet. Once the people start coming in the lab, it starts to get more more noisy, and people start knocking on my door and so on. And so I may write, and then I tend to do less intellectual things, I guess I would say, as the day wears on, because it gets busier and so it's harder to concentrate.
Hmm. No, that makes a lot of sense. Someone called Eloise has written into the chat box. Can you share a moment in your career when you thought this is incredible?
Yeah. Um, the my our aha moment, my aha
Yes,
was when we first did the first database search,
I can
and
imagine.
it worked,
Hmm.
and it was like, this is going to be transformational, because one of the when I left graduate school, so when I was in graduate school, I was interpreting all these tandem mass spectra by hand, and I was sequencing single proteins, and I still filled up a garbage can, about 55 gallons, with uninterpreted mass spectra. And so it was clear to me that we had to come up with a better way of dealing with the spectrum. And one of the things I initially started working on was what we call de novo interpretation.
Hmm.
So just take a spectrum and try to come up with a sequence off of it. And that was not very accurate, because 40 years ago, 35 years ago, the mass spectra were not super high quality
Hmm.
like they are now. So 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 it turned out that, 1994, 1993, the quality of the data was fine for doing database searching, and so that was a real super aha moment for us.
I
And it
bet.
really opened up a lot of possibilities. So not only just sequencing single proteins, but also this ability to take protein mixtures and identify, because tandem mass spectrometry is really good at sequencing mixtures,
Hmm.
or characterizing molecules and mixtures. That's its strength.
Fantastic. Um, Wonderful answer to that question. We've just had an absolute flurry of questions coming in. Let's see if we can pick one here. Someone has just asked, is there any way to link the alterations in proteins to the gene expression datasets and find causes for certain diseases?
So there's some perspective here.
Hmm.
So geneticists think that all diseases you can trace back to a gene or a bad gene or something
Hmm.
like that, and it's that's probably not true, and that there are, like, if you have there are two genes that indicate very high risk for Alzheimer's disease. This
Hmm.
ApoE4 gene. So if you're homozygous for ApoE4, you have a very high risk um, of getting Alzheimer's disease,
Hmm.
but the risk isn't 100%. Um, so it could be if you live long enough, I guess, but there are other things that come into play: environment, diet, uh, other kinds of risk factors. You smoke, you drink, all those kind of things that come into play, and you could think about those um, playing a bigger role on proteins
Hmm.
than than you might on genes themselves. So so there is a belief, particularly in the proteomics field, that proteins and the other component, the environmental components that play around with proteins, may in fact allow you to better understand the disease versus versus the genetics alone.
Hmm.
So, so it's been interesting. So I've been watching the genomics field for 35 years, because
Hmm.
I've been a big fan, uh, but because it helped. I mean, it made my career in a sense. But one of the things that I've found interesting is that, when the genome started, it was, we need to sequence the human genome. So they picked one
Yes.
genome and they sequenced it, and then they said, well, all the answers will come. But when they didn't, it was like, oh, we got to sequence 100 more genomes, and
I
and
think
then
I
they
keep
did that.
going.
And so the answers will come, and they didn't. And then so now that was 1,000, then it was 1,000,000, and now we got to sequence everybody. And there've been a lot of these studies called GWAS studies, which are genome wide association studies, and those
Hmm.
help you to try to find um, risk genes or risk mutations or single nucleotide polymorphisms that might be associated with disease risk. And uh, for example, they've done a lot of these studies on Alzheimer's disease, and there are known genes that cause familial Alzheimer's disease, but the the other risk is very low. It's like maybe 50% or something like that. It's not super high, and so there's been this belief in the genomics field that there's missing heritability, which is what they call it. So we need to do more studies. We need to do GWAS studies on a million more people, things like that. The numbers just keep getting bigger and bigger and bigger. And it's like maybe the missing heritability is related to the other molecules that are involved in cells, like proteins and
Yeah.
metabolites and lipids and things like that.
Hmm.
But that's not what they do. They do genomics, and so their first inclination is to do more genomics.
Makes sense to them, I guess. Fantastic. So we've also had some more questions here saying, do you see a future where top-down proteomics becomes much more common for studying intact protein complexes and their modifications?
Sure. Yes. The only thing holding back that approach
Hmm.
is simply the technology.
Yes.
So proteins. So one of the first experiments I did when I got an electrospray ionization mass spectrometer is I took an E. coli digest, E. coli lysate,
Okay.
intact proteins,
Hmm.
and I tried to inject it in the column and then run it into the mass spectrometer and try to measure the intact molecular weights of all the proteins in E. coli, and it promptly clogged the column. And so I said, well, I guess this isn't going to work. So I will. There's a lot of naivete and associated with that particular experiment. But then I thought, well, I guess we're going to have to digest up the proteins and turn them into peptides to make them more soluble to go into
Hmm.
the column and stay in the column. Proteins are far more difficult to work with than peptides, and that's why the vast field of proteomics uses peptides uses digested proteins rather than intact proteins, because the technologies, the separation technologies, the mass spectrometry technologies are just not as mature and as robust as they are for peptides.
Hmm.
But it's coming. They're making progress.
Hmm.
There are now mass spectrometers which are much more capable of analyzing intact proteins. And so it's coming along, which is why I've jumped in.
It's on the horizon. Fantastic. Let's have a look for some more questions. I've got one here that says, how do you view the potential of in situ proteomics for studying protein localization and functioning in living cells and tissues?
So living cells and tissues. So spatial proteomics is now become a really hot field, and it uses
Yes.
mostly multi-imaging, some of these imaging techniques. So there's also
Hmm.
a method called CyTOF that uses uh, antibodies in a gun, ion gun to let's say a gun, an ion gun to uh, to ionize the molecules.
Hmm.
But spatial is moving along nicely. A lot of it is looking at metabolites rather than proteins. Proteins are a little harder to do
Hmm.
by at least by MALDI imaging. Uh, there are dynamic range issues and all other sorts of things. The MIBI technique, the CyTOF, works better on intact proteins.
Hmm.
But you're just analyzing. You're not really analyzing the intact protein. You're analyzing an antibody that has metal tags on it. So you're analyzing the metal tags,
Hmm.
but it still gives you very nice spatial information that you can't get by other means. So it's coming along.
Nice.
It's a hot area right now.
It's a hot topic. You're not
Yeah.
wrong. So let's have a look. We've got another question here that's saying, well, there's actually quite a lot of chat, John, actually, about personalized proteomics, especially in healthcare settings. So I'm wondering if I summarize all these questions and ask you kind of what are the potential benefits of personalized proteomics in healthcare? Or do you see that being sort of a possibility on the horizon?
Uh, yes. So it although she's become a bit of a, a pariah. So Elizabeth
Hmm.
Holmes and Theranos had that
Yes.
concept of consumer medicine, where you would pop into the local drugstore, give a sample of blood,
Hmm.
and you'd get back a profile that would tell you how you're doing. And right now, at least in the U.S., I go to see my general physician once a year. I get some blood work done,
Hmm.
and so it's a snapshot in time. Now imagine you did that once a month, or you did that every couple of weeks.
Yeah.
If you had the ability to do that, you could you could make course corrections in your lifestyle
Mm.
on a regular basis, rather than going once a year and having the doctor say, well, lay off those Big Macs. That's not good for your cholesterol. So
Yeah.
if you had the ability to do this on a more regular basis, that might be very useful for people to make
Hmm.
course corrections on their lifestyles.
Yeah.
And there have been some efforts to do this. I think Mike Snyder's probably got a company that does this. Lee Hood started one.
Hmm.
Craig Ventnor started one. They were all geared towards rich people that could afford
Afford it.
concierge. They were was basically concierge medicine.
Hmm.
Uh, but once it's like the first Teslas that came out were super expensive, and that helps fund
Yep.
the
The
and
rest of
drive the
them.
technology, so it becomes lower cost.
Hmm.
So if you have this kind of personalized medicine and rich people helping to fund the technology and fund the um, bringing the driving the cost down, then it starts to benefit everybody. But
Yeah.
they kind of all fizzled out. I guess there weren't enough rich people that were interested in doing
That we're interested in the health.
it. So so yeah, I think there would be some benefit to it. It's going to take
Mm.
a lot of study to get there. I don't think it's
Hmm.
coming next year, uh,
Yeah.
but
It's a way
maybe in
off.
time.
Yeah,
I mean, it's still
absolutely.
expensive to do these sorts of things, and there was always this joke. This may have changed now. Is it may cost you $1,000 to sequence your genome, but it costs $1 million to interpret it.
It's a good one. So we've got a kind of applications sort of question here from Paul, asking for your thoughts on technologies on the horizon for high throughput proteomics, if you know of any newer technologies coming through.
That sounds like it could be a stealth marketing question.
Doesn't it a little
Yeah.
bit?
So one of the areas that so mass spec based proteomics works incredibly well everywhere but blood,
Hmm.
and it's improving in blood, but it's not it doesn't have the kind of throughput that you might want if you were going to do a study of 1,000 people or 10,000 people or something of that nature.
Hmm.
And so there are some other technologies that are coming along. There's a company called Olink, which has an antibody-based
Yeah.
system for doing this.
Hmm.
There's another company called SomaLogic that has aptamer-based system for doing this, and then there's a company called Quantum Sci. I'm not sure if they have the necessary throughput, but they are a
Hmm.
different technology for identifying and studying proteins.
Hmm.
So, and there's another company, Nautilus Biotechnology.
Yeah.
So there are a bunch of startups that have come along. One of the ones that I've always been fascinated with, because I actually did Edmund sequencing when I was in the Hood lab, is a company called Ariason that came out of Ed Marcotte's lab that uses um, they've renamed as fluoro sequencing,
Okay.
and that's kind of cool.
Mm.
It has I guess it has potential for a large scale, but the company still
We'll
doesn't have
see.
a product yet.
Mm. They're very nice. I think
So
it.
so there are technologies out there that are going to challenge mass spectrometry, particularly in that space
Mhm.
of serum and blood.
Yeah, absolutely. So just to remind everyone, we've got about 15 minutes left for this session, and do make sure to pop your questions into the box on the right-hand side of your screen and click send. And I will be asking as many as possible in the time that we have available. So a really, really nice one I've come across here is: have any of your proteomics research findings affected your everyday life?
No,
No, short and sharp.
no. I mean, they're they affect me in the sense that when we discover something, it's always really super exciting. And
Exciting. Hmm.
yeah, because most of science is about um, fail. Sorry to say, but failure.
Yeah.
And so the experiments that don't work, or hypotheses that don't work out, or something of that nature. And when something works, uh, it's really exciting.
Yeah. No, I absolutely agree. I think another question that I have personally that I always ask people on these Ask Me Anything sessions is, what do you think are the most kind of crucial areas of focus for future development in the proteomics field, in your opinion?
Well, I think I just told you three.
Well, you kind of did.
So people have been so single cell proteomics is right now is kind of is a fairly hot area, and
Hmm.
people have been toying with this area for a long time, going back
Hmm.
into the uh, probably early 1990s. Sometimes they would just analyze like a red blood cell, and that's kind
Hmm.
of a trick experiment in a sense because there are two major proteins in there, hemoglobin and carbonic anhydrase.
Hmm.
And so they would see those and they would say, oh, we did single cell analysis. But really wasn't all that useful because you couldn't extend it. And there, back in 2018, Nikolai Slavov's lab um, came up with this TMT based strategy to look at single mammalian cells. And now that was interesting. So the mass spectrometers had gotten capable enough that you can now look at a single mammalian cell. And so that kind of spurred this revolution and drive to try to improve on single cell analysis, and it's getting better.
Hmm.
And there are a couple of reasons why you might there are a few really, I think, killer applications of single cell analysis. So one is if you've got, say, a solid tumor and you
Okay.
want to know how the cells are evolving from the starting point in the center,
Hmm.
you could look at each of the cells, the single cells along the way, and you can see what kind of changes are emerging on the proteome level. So you can also sequence them, and you can find all these mutations in the genome and so forth. But which ones are important? Which ones are helping to drive those tumors? And so you want to look at the proteins as well, and you also want to look at the modifications. And so that's technology that's coming along, and that's going to lead to, I think, a lot of really interesting insights into uh, how. I mean, you can look at the evolution of a tumor as a series of natural
Hmm.
selection, and so it's the collections of mutations that are occurring in these cells that are creating cells which are more fit to propagate and
Hmm.
create the tumor. And a lot of what happens as tumors are growing is that you get cell death because they make the wrong mutation. The wrong mutation occurs, and it knocks out an essential protein, and all of a sudden the cell dies.
Hmm.
So being able to study that at the single cell level, I think, will lead to really interesting insights into how tumors propagate, how they stay alive, what are the drivers.
Massively. So we have a question here who someone is asking, how can proteomics data be used for drug safety predictions?
Ah, so one of the early applications of 2D gel electrophoresis was just that.
Okay.
So you would feed a drug to a rodent model,
Hmm.
and then harvest the liver, and then run the liver in a 2D gel. And if you would see certain types of protein signatures, things in particular that were bad were like peroxisome proliferators, then you would know that this was a drug that maybe not doesn't merit further investigation.
Hmm.
So that was being done by Lee Anderson. He had a company called Large Scale Biology to try to do this. He had a tough time getting um, adoption by pharma for some reason
Okay.
of the strategies that he had developed. But certainly one of the hot areas in proteomics is what we call chemical proteomics, and so that's more along the line of the drug and target discovery realm.
Yeah.
But you can also use this to look for drugs that might be uh, bad. So we have a paper we're about to submit where we've developed some methods to look for off target effects of drugs,
Hmm.
and and that those are important because that's where the side effects often come from.
Massively.
It, so lots of really interesting things you can do with proteomics.
Nice. A little bit of a teaser of a paper there. Someone has also just said they've been listening in right from the start, and you mentioned that you did a little bit in neuroscience, and they just wanted to ask you a little bit more about that.
A little bit in what?
About neuroscience, you mentioned right at the
Oh,
beginning that you'd
neuroscience.
done a bit of neuroscience.
Yeah, so I think one of the other killer applications of single cell proteomics is looking at neurons,
Hmm.
and so uh, behaviors and functions in the brain are often driven by small circuits, and so understanding what are the molecular changes going on in those small circuits can be really interesting. Um, we just had a paper with a colleague at Scripps on single cell neurobiology,
Hmm.
and he was looking at um, it's known that certain mutations that are associated with Alzheimer's disease cause hyperexcitability in neurons,
Hmm.
and that there's some belief that this hyperexcitability can be something that causes neuronal exhaustion, causes the neurons to die.
Okay.
And so he engineered um, using iPSC cells, stem cells, a neuron that had just a single mutation in the presenilin gene, going from methionine to a valine, and it converted a normal neuron into this hyperexcitable neuron that's similar to what you would see in Alzheimer's patients. And so, if you could identify the molecular mechanisms behind that process and then come up with a drug that
Hmm.
would tamp down that process, you might be able to limit the hypothesis is you might be able to limit the neuronal death as people start progress through Alzheimer's.
Yeah, which would be pretty interesting.
Yeah, and it's very difficult to it would be difficult to study that at the bulk level,
Mm,
because not all neurons are going to be hyperexcitable.
okay.
And so we have the ability. So one of the things that we've done in this system is use patch clamping to find patch clamping electrophysiology,
Okay.
where we measure the electrical currents coming out of the neuron to find the neurons which are hyperexcitable. We then collect that specific neuron and we do proteomics on it. So we don't look at the ones which are not hyperexcitable.
Nice. Okay, cool. I really think that's pretty interesting. So we have about 5-ish minutes left to go. And someone here has just asked a question, saying, what significant advancements do you see coming up in clinical mass spec over the next decade or so?
So clinical mass spectrometry has been it's been percolating along. So everybody has expected this to be the next $1 billion market,
Hmm.
and it's been slow to come along. And and I but there are some laboratory companies like Quest and LabCorp
Hmm.
where you think that um, you don't realize that a lot of the tests that they provide are coming out of mass spectrometers, and particularly the doctors. Sometimes I've gotten a review back on a clinical diagnostic paper where we've used mass spectrometry, and the reviewer says, mass spectrometry in the clinic, that'll never happen. It's like, you don't know where your hamburger is coming from. Because you order up that vitamin D test, that's coming from a mass spectrometer.
Yeah, absolutely.
Yeah, and there are tests for insulin, IFG, insulin growth factor 1, that are based on mass spectrometers, and so there are all kinds of tests that are currently being done. But people
Hmm.
expect more, and there are people trying to drive this forward. And in fact, if you want to dive deeper into clinical diagnostics, a friend of mine up at Cedars-Sinai, Jennifer Van Eyck,
Hmm.
has a huge operation trying to to both develop diagnostic tests, but also to deliver them to physicians at her medical institution. So she'd be a good person to talk to if that's the
Mm,
direction that you want to go with on Ask Me Anything. Um, but yeah, it's coming. It's
it's on the
just
horizon.
it's just moving very slowly.
Snail's pace. So we have also had a lot of people in the comments saying if you have any advice for students interested in entering the field of proteomics, they just want to hear your advice, really.
Yeah. So when I started, I was more mass spectrometrist than biologist,
Hmm.
and I think the current trend is people are more biologists and then learning the mass spectrometry, learning how to use it.
Hmm.
You don't necessarily have to know how to build mass specs. So when I started, we were still building mass specs in the laboratory. But now you can just buy them. So, but learning how to what the data means, how to interpret the data, and all that kind of thing. So a lot of the people in my lab, when they arrived as to do postdocs, had no idea how to use a mass spectrometer or how to interpret the data, and they've all learned that, and they came from neurobiology fields and cell biology fields and so forth. And so I think it's to combine those expertise. So maybe you do your graduate work in a biological study, and then you go do a postdoc in a lab that has access or mass spec capabilities to learn that part of that that part of the equation. So you
Mm.
combine those two things together to study with more um, tenacity, deeper understanding of a particular disease.
No, absolutely, and bringing it all together. Fantastic. So we only have a couple of minutes left, but one of the questions, again, I love to end and ask me anything on, is John, where do you see the future of proteomics going, or where would you like to see the future of proteomics going in the next 10 years?
So you get a different answer depending on who you ask for this question. But I kind of gave my my vision for where I think proteomics should go based on what we are doing. I
Hmm.
mean, it would be kind of silly for me to say, "Oh, you
To say
do this,"
not.
when we're doing that. But I think it's single cell. I think it's conformational structural proteomics. So understanding the shape of proteins within the context of a cell,
Hmm.
and that was all driven for us by a project, long running project we've had in the lab looking at cystic fibrosis. And so we got to the point where we were there was no when we were asking these questions there was no structure for the CFTR protein.
Hmm.
And so we were asking questions: what's this thing look like inside the cell? And so we're trying to come up with a way to look at it, and we developed a strategy that allowed us to do this chemical footprinting study. We actually found what we think is a critical conformational interaction within the protein that the cryo-EM people missed because they had to cut out the section of the protein that contains that interaction.
Hmm.
So we think we found using this method and looking at inside the cell, we think we've found this critical interaction that decides whether the protein is mature or not mature, whether it gets destroyed or gets out to the cell surface to do its job. And that was all based on asking that kind of question. And then the third thing is this being able to look at proteoforms and trying to look at proteoforms. What I really would like to be able to do is to take protein complexes, use native mass spectrometry to measure that complex, and then start tearing the components apart and looking at the proteoforms of each of the components. So I think that would be being able to do that in a robust and high throughput way would be fantastic.
Let's
That
hope
probably won't happen
that's.
in my career time.
We can have our fingers crossed in the next 10 years. That can happen. Perfect. Thank you ever so much for joining us here today, John. Unfortunately, due to time constraints, we must conclude the session, but it's been an absolute pleasure speaking with you. And based on the comments, everyone else has loved it as well. So thank you ever so much for your time.
Oh, great. Thanks. This was fun.
Perfect. And before everyone goes, we'd love to hear from you. Did you enjoy the session? Please let us know with the form that will appear shortly on your screen. This AMA will now be made available on demand. So if you have any friends, family, or colleagues who you think might have been interested in this discussion, please feel free to share the link with them. And once again, thank you all for your time and for your participation.
Have a good day.